All women are at risk for gynecologic cancers, which account approximately for 12% of cancer diagnoses in the United States. We hypothesize that there is a relationship between the biological pathways that characterize gynecological carcinomas and that it is possible to gain further insight into the pathogenesis of these diseases by constructing a trait pathway network. We decided to use the trait pathway analysis since this one takes into consideration the relationship between the genes instead of only the genes in general. For the data, we use the GWAS catalog, which is known as genome-wide association studies, and it's used to see if any variant is associated with a trait, centering its search on association between single nucleotide polymorphisms and traits. We use its summary statistics from 14 studies looking at a gynecological carcinoma phenotype being breast, cervical, endometrial, or ovarian. All studies included individuals of European ancestry. For the pathway analysis, we ran Paris. Paris takes the results from our GWAS summary statistics and looks to see if associations in a pathway are more than you would expect by chance, given the number of genes. We use its summary statistics at a discovery p-value threshold of p1e to the negative 4 for each study. We then perform 10 replications of Paris for each phenotype and consider a pathway to be a significant association with the phenotype if it appeared as a hit in 5 or more replications. The LD blots are used to group the SNPs into LD features, and they were calculated using the individuals of the European ancestries from 1,000 genomes, and the pathway features were extracted from the KEGG database. We then created a trait pathway network based on the results from the Paris analysis, where each edge is a significant association between a cancer phenotype and a KEGG pathway. In Figure 1, we have a phenogram of mapped genes associated with gynecological carcinomas. We used SNPs from the GWAS catalog summary statistics, and they were mapped to genes based on a GWAS p-value threshold of p5e to the negative 8. In this, we were looking to see the pleiotropy in each gene and phenotype or trait. The pleiotropy is known as one gene determining multiple traits. And as you can see, we didn't see as much pleiotropy as we would have liked, since most cancer traits, cancer phenotypes, are mapped to only one gene and they don't have as much pleiotropy. This is why we decided to look for significant associations per study, which is shown in Table 1. In this, we discovered that there were 156 significant associations. We used that data to construct the trait pathway network of gynecological carcinomas in Figure 2. In this, each node is a phenotype or pathway, and each edge is a significant association between phenotype and pathway. Instead of looking at pleiotropy, we were looking at what we refer to as pathway-based pleiotropy. This is because, since I explained before, the pleiotropy is known as one gene determining multiple traits. In the pathway, we were looking for shared biological effect of each pathway with the genes. In this, we did see much more pleiotropy, pathway-based pleiotropy, backing up our hypothesis. Despite lack of evidence of pleiotropy at the SNP level, several human trait pathways such as type 1 diabetes, systematic lupus erythematosus, and autoimmune thyroid disease were associated with both cervical and endometrial cancer, supporting previously known relationships and highlighting the utility of a pathway-based approach in understanding the etiology of these cancer types. Future directions include expanding the analysis to include comorbidities of gynecological cancers, such as endometriosis and heart diseases, and constructing similar trait pathway networks in non-European populations to identify ancestry-specific differences in disease etiology.